Okay, this PowerPoint correlates with Mendelian Genetics, Part 5. This is notes page 27B. All right, for important concepts, we know that there can be not just chromosomal errors, but gene errors. And we've talked about point mutation and reading frame shift mutations previously. So, the point mutation is where you have one nucleotide that gets replaced by the incorrect one. And it's just there on that specific point. It can be silent if the same amino acid that was supposed to be there is brought in, or it can cause problems if it brings in another amino acid. And then reading frame shift mutations are those where a nucleotide has been deleted or inserted, and so everything kind of shifts downward. All right, so what we're talking about here is chromosomal errors that occur. These can occur during mitosis or meiosis. They would occur during anaphase most often when the chromosomes are moving around, or during crossover during prophase 1 when you have segments, DNA segments that are exchanging places. So when we're looking here, you can see that with non-disjunction, where they're not separating correctly, all right, you've got during anaphase in meiosis 1, when they're supposed to move apart and away from the middle, you have this pair that stays together. Okay, so when it is supposed to be separating out, you're going to wind up at the end with a couple of cells that have an extra chromosome. These are all supposed to be diploid, so they're supposed to have 2 in each. Right here, but instead they only have 1, and then you have a couple with 3. Okay, so this shows you if it happens in meiosis 2, in that second anaphase, where they're supposed to separate out, that individual chromosome is supposed to split into sister chromatids. It's supposed to split like this, but instead that one stays together. You wind up with 2 cells that are normal. They're supposed to be diploid, have 2 chromosomes there, but you wind up with this one has an extra and this one is lacking one, because here this chromosome stayed together instead of splitting out into sister chromatids. So this is what we call non-disjunction. So write yourself a note on the 2 types of errors that occur. The chromosomal number, where the number of chromosomes is actually messed up. Put there that cells should be diploid. They should have 2 of each kind of chromosome, 1 that they got from their mother, 1 that they got from their father. And instead you can have aneuploidy, which means that you have an abnormal number of chromosomes. Okay, so you can have trisomic problems, and this is where you have 3 of each kind of chromosome, as in the case of this first one right here. Okay, so that's a trisomic problem. You have 3. Tri means 3, so underline tri there in your notes. All right, or you can have a monosomic problem, and mono means 1. Okay, so we've got 1 chromosome as we do here instead of having 2. Okay, you can also have polyploidy. Poly means many, and this is where you just have lots of extra entire sets of chromosomes. Okay, so to show you what I'm talking about, you can have triploid. And remember, if N means the number of individual chromosomes that we have, the number of types of chromosomes that we have, in a human that would be 23. Instead of 23 chromosomes, you have 3N. So you have 3 of each kind of chromosome. So this is called triploid. When it says 3 halves, it's talking about 3 sister chromatids of each kind of chromosomes. Now, these plants, these can often happen in plants. In fact, we genetically engineer those to happen that way, because typically in triploidy, the plants are going to be sterile. They actually don't even have seeds inside them. So if you've ever had a seedless watermelon, you had a genetically engineered triploid organism. So they'll do that, arrange it so that one of the chromosomes does not split during anaphase, and you wind up with, at all, and you wind up with 3 of every kind of chromosome. Now here, this is a human karyotype. Organisms, or humans, and generally animals that are conceived with triploids, with 3 of each kind of chromosome, do not survive. Now, it's not that unusual in plants. Like I said, we can genetically manipulate those. Seedless bananas and watermelons are all triploid, but typically that's not going to occur in animals, okay, or humans. All right, then you can have tetraploid. Tetra means 4. And so you have 4 of every kind of chromosome. One thing that we have genetically engineered to do that is ryegrass. For whatever reason, it tastes sweeter, and livestock actually prefer the taste of tetraploid ryegrass as opposed to diploid ryegrass. And so these two cells are not allowed to separate at all when they come together, so the gametes. So you wind up with 4, a full set from both gametes. They just all stay together, so you get tetraploid instead of diploid. Okay, so 4 of each kind. Now, that's one kind. The chromosome number can be messed up. Individual chromosome structure can also be an issue with an error in chromosomes. So it says in your notes, try to see the terms, and you can see this as we go along, what the notes are talking about there. But these occur in prophase 1 because crossover messed up. Okay, so in prophase 1, where pieces of your chromosomes trade places, this was faulty. It did not happen correctly. So you can have deletion. So this is where a chromosome segment actually gets deleted. So right here you see D. It is not present here. So during crossover, it actually just got pulled off and deleted entirely instead of trading places. So it got missing, or went missing. Basically, it gets stuck on the other chromosome. Okay, you can have duplication. And duplication is where you have a piece of a chromosome here, okay, B and C, and it winds up being duplicated. It's there twice on the chromosome instead. So duplication just means you have the same thing 2 times. It winds up missing from the other homologous chromosome. Both of them get put on the same one. All right, then you can have inversion. Now, to invert something means that you turn it backwards. So you can see that here, when BCD is supposed to cross over, the information that it got put in backwards, and it's DCB. So inversion just means that during crossover it got flipped upside down when it traded places and wound up being put in there backwards. And then translocation is the last kind of individual chromosome, the structural deformity. And that means that your chromosome segment, instead of crossing over to a homologous chromosome, to the same kind of chromosome, it actually goes to a different autosome, one of those number 1 through 22s. It goes through it to a different chromosome altogether. So it doesn't attach to the homologous pair. It winds up going to the wrong one entirely. Okay, so those are examples of individual chromosome structural problems. Now, trisomy 21 is often referred to as Down syndrome. Okay, so if you look at this is actually a disorder that's caused by an abnormal chromosome number. Okay, so it affects about 1 in every 700 births. And the problem is that they have an extra number 21 autosome. Okay, remember again, our autosomes are numbers 1 through 22. The 23rd pair right here is called our sex chromosome. But this is a problem with an autosome number 21, and that is why it is called which means 3, somy 21. You have 3 number 21 chromosomes. So Down syndrome is not that terribly uncommon. Some of the symptoms that are generally present are there's cognitive delay, so they may learn more slowly. They have generally extra adipose tissue around their eyes that gives them an appearance of squinted eyes, and more of a flattened facial appearance. In women, this is more common in women who have children after they are 35 years old. And that is because, remember, females are born with every egg that they're going to have. So when the woman is 35 years old, her eggs are also 35 years old. And so the spindle fibers weaken after fertilization, and during anaphase 2, those spindle fibers don't hold on to the chromosomes like they're supposed to because they're old. And so they don't release one of those, that number 21 chromosome, like is needed during anaphase 2. And so you wind up with an extra, and you wind up with 3 chromosomes instead of only 2 number 21s. All right, now extranuclear DNA is mentioned there at the end of your notes. Extra means beyond or outside, okay? So extranuclear DNA is DNA that we know is not, we've already talked about it, it's not only in the nucleus. You've got DNA in your mitochondria, in your chloroplast, and you also have them in the plastids as well. Now, mitochondria gets inherited from the mother inside of her egg. So she has the mitochondria inside the egg, and that's where those mitochondria come from. And we know, we've talked about before, that in forensics, in criminal forensics, they often use mitochondrial DNA whenever they're trying to show a relationship between individuals. So in mitochondrial myopathy, this is where the DNA is faulty inside the mitochondria. And these mitochondria don't really have the ability to make a lot of ATP because the DNA is messed up. So since they're not able to make energy, it would make sense then that these individuals with mitochondrial myopathy have a lot of fatigue. They're very tired all the time because they're not able to make an adequate amount of energy